Hello, this is Insang Lee. Last week we discussed about that how can we develop the new radiopharmaceutical, and today we will focus on a specific radiopharmaceutical that can be used in a clinical practice. So, but I think also that a compound that radiopharmaceutical is under development for clinical use. So first of all, we start with the technetium radiopharmaceutical because of its specific feature of the technetium is the very good for gamma camera and also sodium iodide scintillator, and most of all that technetium 99M can be generated from the technetium generator already mentioned last week. And this technetium generator was developed by the Korean domestic company Samyang Unitech, and that price is almost half of the imported generators. Because of the half-life and also gamma energy is good for imaging study, so technetium was developed. So many different types of technetium radiopharmaceutical already developed and also used in clinical practice. As you can see here, technetium can emit the gamma ray that half-life is around 6 hours. And also, I already mentioned that the cold kit can be used for technetium radiopharmaceuticals. So already developed with this convenient labeled kit that possess that ligand such as DTPA, HMPAO, or MAA, or any other kinds of chelating agent. And also that kit contains tin chloride as a reducing agent for from technetium 99M O4-. That chemical species can be reduced into technetium 5. The charge is 5+ or 3+ or 1+. And also it that after the radio labeling, that radiopharmaceutical should be stabilized by the ascorbic acid or gemcitabine acid or any other core factors for core chemicals. Technetium pertechnetate can be used as a imaging agent for thyroid or stoma. As you can see here, that tetrahedral structure of the technetium O4-, that is almost the same structure with the iodine, radioiodine, and also that possesses that minus charge. And that size of technetium O4 is almost same with the iodide. So that's why that technetium O4 can be uptaken by the thyroid and also stoma. Last week we discussed about that colloidal structure of radiopharmaceutical. Here you can see here technetium MAA and another colloidal structure, especially antimony sulfur colloid can be used as a lymphatic flow imaging because of that size is around the 3 to 10 nanometer. That is good penetration into the lymphatic pathway. Here you can see the technetium MAA macro aggregated albumin can block the microvessel in the lung. So you can visualize that the function of the lungs. So if that blood vessel was blocked and that decreased uptake of technetium MAA, that means that that lung cannot act as their own function. So that's why technetium MAA can be used as a lung scan agent. Sulfur colloidal structure can be uptaken by the liver, especially for from the corpuscle cell in the liver. So if that patient has a chronic fatigue, and then if you inject into that patient, you can see the slight decrease uptake of the sulfur colloid. That means that that liver function has a problem, and you can see where is the foci of the problem occurring in the liver. And antimony sulfur colloid, as I mentioned, that size of the colloidal structure is very small. So that's why after the injection of that technetium labeled antimony сс is passed through the lymphatic pathway, and also you can see the lymph system, and also sometimes you can find the lymph node in the human. And another application of the antimony sulfur colloid, technetium antimony sulfur colloid, is a sentinel lymph node detection. Especially for a breast cancer patient, that original site, primary site of the cancer will be removed from the breast, but still there are some possibilities of the metastasis of the tumor cells. So if you find the sentinel lymph node, and then you dissect the sentinel lymph node, and then you can see in the microscope, and if you find the cancer cell in that lymph node, then you can dissect all the lymphatic pathway and or the lymph node from the near to the breast. So earlier we tried to make technetium antimony sulfur colloidal structure used for a breast cancer patient, sentinel lymph node detection. But now we focused on the monocylated serum albumin labeled with the technetium 99M. Here you can see the technetium MDP structure, and also HDP is the some derivatization of the from the MDP. Mainly that the bone structure is that contains that the pyrophosphate structure. So that's why that MDP is the mimic of the pyrophosphate. Actually, the technetium labeled complex, that's the structure of the technetium labeled MDP complex is not clear till now, but still that MDP technetium MDP can visualize that bone structure. Here you can see clearly show the bone uptake of the technetium MDP. And another application is the kidney imaging. with the technetium DTPA or technetium mercury. As I mentioned last week, technetium labeled minus charged compound can visualize the kidney because of that. Kidney excretion rate is very high than any other compound. So you can see that technetium DTPA and technetium mercury is the major radiopharmaceutical used for kidney imaging. You can see how that technetium DTPA can be excreted via kidneys. So you can see the increasing uptake in the bladder, and you can see which kidney has a problem or not. And sometimes we can use the probenecid as a that kidney blocking agent, and then you can see the high concentration is still remained in the kidneys. And another technetium labeled compound is for hepatobiliary agent. That is a highly lipophilic compound. So that highly lipophilic compound can be uptaken by the hepatocyte in the liver. So that liver, after injection of that technetium derivatives, and then you can see after some time that all the radioactivity in the liver was cleared, and then you can see the high gallbladder uptake and then also intestinal uptake after the technetium injection. So this means that liver function is good with this patient. Another radiopharmaceutical is the technetium HMPAO or ECD as a brain perfusion imaging agent. As you know that if that brain vessel in the brain was ruptured or blocked, and then that you can see the lower uptake in the brain with the injection of the technetium HMPAO or ECD, and then you can vascularization in the brain, and then after the injection of this compound, and then you can focus on which part is the blockage of that brain vessels. Here is some different types of brain perfusion imaging agent, iodine 123 labeled IMP, and also HMPAO and ECD. That maximum uptake ratio and time is different, but almost the same retention in up to 90 minutes, or sometimes 10 minutes or 20 minutes. And that uptake mechanism is slightly different, but that function of these kinds of different types of radiopharmaceuticals is almost the same. So we've selected technetium HMPAO because of that convenience of the technetium labeling. Another type of technetium labeled compound is myocardial imaging agent. Here you can see the technetium MIBI structure that suggests that the highly lipophilic and also positively charged, especially ether group is inside of the structure. Because of that, these types of ether can be cleaved in the myocardial cells, and then that can be after the cleavage of this methyl group, and then it forms an alcohol that hydrophilicity is increased, and then that remains in the myocyte. And also we can do the technetium MIBI with the rest or stress imaging. Exercise should be done in the treadmill or bicycle, and sometimes adenosine or dopamine stressed imaging. So sometimes we can do these kinds of reconstructed images, that is quantified gated myocardial SPECT image. Then you can see the lower uptake in the myocardium. Then we can get the information from these images of the which kinds of vessel was blocked or has a leakage. And we can label technetium in for blood component. So especially red blood cell RBC labeled with the technetium HMPAO, and then after the labeling of the RBC, then we re-inject into that patient, and then you can see the blood vessel in the human body. Here you can see the how can we label with the RBC with the technetium compound. Especially we use the technetium HMPAO as a RBC labeling. For RBC labeling, you can withdraw withdraw the blood from the one specific patient, and then we can label with the technetium HMPAO, and then we centrifuge, and supernatant was removed, and then that lower plaque was resuspended and injected into the that patient, and then you can see the blood vessels with the administration of technetium 99 and RBC. Another example is the white blood cell is labeled with the technetium 99N. That technetium 99N WBCs can show the which site is inflamed or not. So this is the inflammation imaging with the technetium WBC scan. Just same with the technetium RBC, we can label it with the technetium WBC, and then we inject that technetium labeled the WBC into that patient, original patient. For development of the protein, especially for a protein, technetium labeled protein, such as antibody or biologically active protein drugs. Here you can see the direct. There are two types of labeling methods: direct labeling or indirect labeling. So direct labeling can be occurred by the small modification of the that protein using beta-mercaptan ethanol or thin chloride or ascorbic acid. And indirect method, after the conjugation of this chelating agent into the protein, and then the technetium 99N was labeled into that chelating agent. Here is a typical example of the direct labeling of the technetium into the antibody. That disulfide bond was reduced with the mercaptan ethanol, and then technetium 99M is that combined with the weak chelator such as MDP or any other weak chelators, and then that technetium is labeled into that thiol groups in the antibody. Here you can see the several different types of the technetium labeling chelating agent, especially DTPA or cyclic DTPA is the first generation of the technetium labeling compound, and another chelator is the N2S2 diamino dithiol groups is there, and also N3S, sometimes triamino or one thiol groups, and hydrogenonicotinic acid. HYNIC is the popular technetium labeling agent, but that the HYNIC group suggests that only 3 coordination number, and then we should use that co-chelator for technetium labeling with the HYNIC group. And as you know that the antibody blood circulation time of the antibody is very high. Even after the injection of the antibody, almost 7 days or 20 days blood half-life in human body. So that's why we should reduce that size of the antibody. So several researchers, they generate engineered antibody, FAB, FAB2, or single chain FB, or any kinds of diabody or minibody. So reduce the size, that means reduce the blood circulation time. So especially for therapeutic radioisotope labeled to these kinds of therapeutic development, for development of the therapeutic radiopharmaceutical, that blood circulation time will harm the bone marrow toxicity, will affect the bone marrow toxicity. So that's why blood circulation time is very important, very major factor for reduce the bone marrow toxicity. So that's why I think that these types of engineered antibody can be used for therapeutic radiopharmaceutical. Here you can see the HCl3 is good for blood circulating antibody. So that is the labeled with the technetium 99M, and also even that the 24 hours, after the 24 hours, you can see the whole body. And especially that the reduced types of IgG can be degraded in the liver. So that's why high liver uptake was shown in these images. So another typical example is the blood pool imaging agent labeled with the technetium 99M into the human serum albumin. After the technetium labeling, you can see the blood vessel in the body, but still high uptake in the liver after only 1 hour after injection, and even in an animal and also human body. So this means that human serum albumin was denaturated, we think that. So in our department, we developed that less denaturated human serum albumin labeled with the technology 99M, and then we can see after 24 hours we can see the high blood vessel images, and after 2 hours you can see the clear blood vessel in my body. And another example is that the application of the radiopharmaceutical is the C11 or F18 compound. That C11 and F18 compound can visualize the brain or tumor or heart, any other functional image of in the human body. Brain imaging agent, especially for receptor or transporter imaging, or sometimes we can use that as amyloid plaque or tau abnormal protein images. And tumor imaging, that mechanism of the tumor imaging is that target for a metabolism or receptor or anti-cancer drug can be labeled with the C11 or F18. Heart imaging, that is the focused on the metabolic imaging or receptor on the myocyte. Here you can see the typical example of the brain receptor or transporter imaging agent. For a dopamine system, there are a huge number of the different types of imaging agents, and also dopamine reuptake site. Earlier, that was called as a transporter. That is the cocaine derivatives, especially F18 CFT or C11 cocaine. For serotonin system, altanserin and catanserin is a famous one, and also metabolic glutamate receptor 5 targeting with the C11 labeled AVP 688. And opioid receptor, that carpentane is famous compound, and also benzodiazepine or cholinergic system can be visualized with the flumazenil or any other CP or dextamide or QNB. In the dopaminergic system, we can see the that dopamine synthesis with the F18 labeled fluorodopa, F-dopa, and also dopamine D2 receptor can be visualized with the C11 raclopride, and also dopamine transport, DAT imaging with the Win derivatives or beta-CIT or TRODAT. So whole system we can visualize that dopaminergic system. Here you can see the C11 labeled raclopride receptor imaging, and this is the normal imaging. And then patient should be reduced that uptake in this specific area of the brain. And then this is the CFT that is for dopamine transporter imaging. So almost same with the receptor imaging with the C11 raclopride, but that resolution is better than the C11 compound because of that F18 that positron from the F18 is that flight range is somewhat smaller than the C11 compound, C11 radionuclide. And here you can see the dopamine synthesis image with the F-dopa, F18 labeled F-dopa. And this is the norepinephrine transporter imaging. But in this image, you can see the high skull uptake. That means the F18 is detached from that original compound. So F18, as you know, that that free F18 can be localized into the bone. So that's why that high bone uptake, that means that detachment of the F18 from the original compound. CL11-DASB is the serotonin transporter imaging. So after the injection of the DASB, so we tried to use the escitalopram as a competitor for CL11-DASB, and we can calculate that the binding potential of the DASB and using escitalopram as a used as a SSRI. So we can calculate what is the optimal concentration of escitalopram for each patient. So we conclude that there are several different types of dose range. So we use the 10 milligram, from 10 milligram to 50 milligram of the escitalopram for treatment for the patient. And here you can see the metabolic glutamate receptor 5 imaging with the C11 ABP 688. And because of that, C11 compound showed a low resolution than the F18. So but that normal distribution in the normal brain. And C11 Verapamil target for P glycoprotein in the brain. After the cyclosporine loaded, that you can see the high BBB image, high uptake in the BBB. Because of that, BBB possesses the P glycoprotein. So because of that kinds of radiopharmaceutical can visualize that the PGP function in the BBB. And F18 flumazenil can visualize the benzodiazepine receptor in the brain. That is the we have to find the foci of the epileptic zone in the brain. So we label the F18 into the flumazenil derivatives, and then you can see the normal brain uptake in the control group. Another application of C11 or F18 compound is amyloid beta plaque imaging. So this is the normal uptake in the white matter, and other this is the image from the patient. So high uptake in the specific region in the brain. After disclosing this image in the one amyloid symposium in the Uppsala in Sweden, after 1 week, science report that these kinds of dramatical imaging from in vivo imaging or using the amyloid plaque imaging agent. C11 labeled PIB is the famous compound for amyloid plaque imaging. So you can see here higher uptake than the normal control. So after coin this kind of structure was developed, and then so many radiochemists devote into this field, and then after 10 years we have a 4 different types of amyloid plaque imaging agent, flutamethasone from G, fluorobetaben from vial shedding, and now I think that is the that compound is, was sold into the Pyjama or any other company, and another compound is the Aljaview from Korean domestic company. That F18 version of that amyloid plaque imaging agent was approved by the US FDA, also Korean FDA. Then now we can cover the health insurance from the government, and also we can, the patient only paid less than $200 for amyloid plaque imaging. This FDDMP, F18 FDDMP was developed by the UCSF. That compound target the amyloid plaque and also tau, abnormal tau protein. But as you know that if we develop some kinds of radiopharmaceutical target only one disease target, is better. Even we can do the amyloid plaque imaging and also tau imaging that can give the more information than the these kinds of complex imaging from the patient. So because of we cannot identify this activity from the amyloid plaque or tau, abnormal tau. So because of these types of compound cannot differentiate that this patient is a tauopathy or amyloid plaque. So these compounds cannot pass through the FDA approval. And then that success of amyloid plaque imaging, that compound is not real correlation with the patient stage. So after that clinical experience, people need to another target for Alzheimer or dementia imaging. That is the tau, abnormal tau protein. So this is the firstly developed by the NIRS in Japan. They developed the PBB3 labeled the C11 compound that can statistically visualize at the abnormal tau in the human brain. And also we set up this compound in our hospital, and then this is normal imaging of the brain. And after that, this version, C11 version of PBB3 also several radiochemists, they want to develop the F18 version of the tau imaging agent. So now Merck or AstraZeneca also focus on the second generation of the tau imaging agent. That is under development. And clinicians in the psychiatry or in our hospital, they said that tau is a better imaging and disease correlation than the amyloid plaque imaging. So I think tau imaging agent will be the better choice for diagnosis of the or disease staging of the dementia, especially for Alzheimer disease. Here is the tumor imaging agent that is related with the metabolism. Yes, you know that the tumor cell is the increased metabolism than the normal tissue. So that each kinds of different types of radiopharmaceuticals accumulated in the tumor cell. Especially F18 FDG can be used as a glucose metabolism, higher glucose metabolism than the normal tissue, and also amino acid for protein synthesis metabolism is also increased in the tumor cell, and DNA synthesis, and then sometimes that that tumor tissue cannot get the oxygen from the blood vessel, so some part of tumor tissue can exist as a hypoxia stage, low oxygen concentration that can be visualized with the F18 labeled meso or F10. And another approach is the angiogenesis imaging with the RGD peptide that is labeled with the F18 or gallium-68, and also especially for tumor-specific receptors such as estrogen receptor that can be visualized with the estrogen derivatives or tamoxifen, and sometimes anti-cancer drug can be labeled with the F18 or C11. So before treatment of these kinds of anti-cancer drug, and then we can visualize that that patient can be treated with these types of anti-cancer drug. F18 FDG is almost same structure with the D-glucose. So that's why that can be uptake to the cells, and then that is accumulated or accumulated into the cell. Here you can compare the D-glucose and F18 FDG. That both compound can penetrate to the cell with the active transporter glucose transporter 1, and then that this hydroxyl group will be phosphorylated with the hexokinase. So in case of glucose, that is the triphosphorylate, that will be the triphosphorylation, and then that is metabolized to the C11, just the CO2. But FDG, after the one monophosphorylation, and then they identified this F18 labeled compound is not the original glucose. So after the monophosphorylation, that FDG monophosphate is located into the cell. So that this concentration is just after the transport into the cell, and then they accumulated more and more in the tumor cell, and then finally you can see the high uptake of the tumor tissue in the human body. So so you can see that these types of imaging in the clinical routine. And another target is the amino acid imaging. So we normally use the C11 labeled methionine as a tumor imaging agent. But as you know that the FDG is a powerful tumor imaging agent. But as you can see, a high brain uptake also occurred using the F18 FDG. So C11 methionine especially we used for brain tumors because of the FDG showed a high uptake in the brain. Then that comparison imaging cannot be occurred in the brain tumor. So that's why C11 methionine as a brain tumor imaging agent. And DNA synthesis imaging, you can see the FLT here, and then FMAU or C11 thymidine. But the FELT is the FDA approved DNA synthesis imaging agent in Korea. Just like a FDG, FLT was accumulated after the diphosphorylated with these types of compound, and then you can see the high uptake in the lung cancer because of the FDG is the best agent for tumor imaging. So that's why that the FDG is sometimes that we cannot identify or differentiate the inflammation or tumor tissue. So that's why FLT is the another alternatives for a lung cancer patient. And hypoxia imaging using the 2-nitroimidazole derivatives that is labeled with F18. That F-myso is that one nitro group in this kind of compound, imidazole compound, and then that is the reduced up to amino. That nitro turns into the amino group, but that is the normally a reversible reaction in the that the oxygen is located into near to this compound. But if that oxygen concentration is low, that that compound should remain in this amino imidazole. So this amino imidazole cannot pass through the cell membrane. So that's why that accumulation mechanism of the F18 F-myso. So in this image, this AB means A means FDG imaging, B means F-myso imaging. So high uptake of FDG, but low uptake of the F-myso means this tumor tissue, that oxygen concentration of this tumor tissue is very high. But you can see the high FDG and high F-myso uptake means this tumor tissue is located in the low oxygen concentration. So if this kinds of imaging was occurred from the patient, that the low oxygen concentration that can affect the chemotherapy or radiotherapy because of that low oxygen concentration affect the tumor specific treatment. So if we use and then we have to increase the dose of the chemotherapeutic or radiological activity into this patient. And I think almost 10 years or two decades, F18 that algae compound can visualize that the new vessel formation, neovascularization in the tumor tissue. So this algae sequence was labeled with the F18 or gallium-6 here or technetium 99M. Here you can see our result of the F18 labeled the algae compound. that ischemic muscle was that the algae compound was highly uptake in the ischemic muscle. Because of that, ischemic muscle should that increase of the alpha V beta 3 integrin in the normal tissue. Here you can see the tumor receptor imaging, especially that is the androgen or estrogen receptor. F18 labeled FES can be used as the EL positive tumors. And now we have approval from the Korean FDA for lung cancer patients in Korea. And here you can see the androgen receptor imaging can show the prostate cancer patient. And anti-cancer drug such as paclitaxel, that is labeled with the F18. Before treatment of paclitaxel, that resistant of that chemotherapy, that you can see either low uptake in the this region, and responsible. We can expect that the responsible for that chemotherapy, and then that uptake is increased in this model. And heart imaging can be visualized with the F18 FDG or C11 acetate or F18 or C11 labeled fatty acid. As you know that our myocardial, myocardial muscle can get the energy from the glucose and fatty acid. And another heart imaging agent is the hydroxyapatidine, that is labeled with the C11, that is the that receptor in the myocyte, on the myocyte. Normally, FDG or ammonia is the gold standard for heart imaging, but radio-labeled fatty acid can be used for heart imaging. Another application is the gallium-68. As I remember that year of 2008, one professor in US, he emphasized that after 10 years, almost 10% of portion of the radiopharmaceutical will be the gallium-68. So nowadays, we use the gallium-68 as an imaging agent for theranostics matched with the lutetium 177 or 1890, especially for neuroendocrine tumors. And also, we developed the gallium-68 labeled PSMA targeting agent that can be matched with the lutetium 177 labeled PSMA targeting agent. Because of the gallium-68 also has a good property for PET imaging. Here you can see the almost 1 hour half-life, and also that is the produced by the germanium gallium generator, and also that half-life of germanium is so long, more than 200 days. So that's why after getting that generator, and then we can use more than 6 months for gallium production. So here you can see the several different types of gallium labeled compound. That is the firstly peptide labeled with the gallium-68 for imaging of the somatostatin receptor, and also alpha MSH, that especially for melanoma, and Bombesin analogs, and also RGD again, and substance P, and also GWEL that is good for the PSMA imaging. And also small molecule can visualize that some specific tumor imaging or myocardial imaging. And sometimes we can label with the antibody, but that gallium half-life is too short to antibody imaging. But in case of us, we labeled the monocylated serum albumin with the gallium-68, and also we can see the lymphatic pathway in human body. And also nanoparticle can be labeled with the gallium-68. Here you can see the neuroendocrine tumor imaging with the gallium derivatives. Here you can see the gallium dotato and dotano, and also that is matched to combination with the lutetium dotatate. But gallium dotato and gallium dotatate has a slightly different biodistribution in same patient. Because of that, somatostatin receptor has several different types, different subtypes. So that's why that affinity to specific receptor is different with these two compounds. So that's why that image is slightly different. But normal combination is gallium dotatate imaging and also lutetium dotatate treatment in the clinical routine nowadays. And alpha MSH analogs can be used as a melanoma imaging. Here you can see clear tumor targeting imaging with the animal model. And Bombesin analog, that is the GLP receptor target, that is the prostate, breast, or pancreatic tumors. And also that was evaluated in the tumor model. And RGD dealt with the gallium-68. Also, that can visualize that the angiogenesis imaging, that the original compound, that affinity of the original compound and NOTA group is almost same. And also in vivo imaging with the animal models. And first in human in Korea, you can see the clear imaging with the high kidney uptake and blood excretion images. And we use this gallium RGD compound as a selection of the AVASTIN responder or non-responder. Here you can see the high liver uptake in the gallium-68 RGD, and then that AVASTIN treatment was treated with the AVASTIN, and then you can see the after some time that AVASTIN acts as a good treatment agent. In the controllee, that non-responder cannot show the gallium RGD image. So that's why we can divide into the non-responder and responder before injection of AVASTIN, before treatment of AVASTIN. And also after some time, we developed the RGD kit for the gallium labeling and then distributed into the several countries in the especially in South America and also Southeastern Asia. And gallium BAFN can be used as a myocardial imaging agent. Here you can see the almost same with the O15 water imaging with the gallium-68 BAFN. And also you can see the high myocardial uptake in the peak. But the first pass extraction rate of gallium bapen is too low, less than 1%. So as I remember that the technetium MIBI is more than 10% of first pass extraction rate. So that's why we dropped this compound as a myocardial imaging agent. And another typical example of the gallium labeled PSMA is for prostate cancer imaging. This is the firstly developed gallium PSMA ligand from the Heidelberg group. That name is a PSMA 11, already listed in the European Pharmacopoeia, and you can see the highly concentrated image in the tumor metastasis from the PSMA positive patient. So just like gallium and lutetium combination in the neuroendocrine tumor, that the prostate cancer treatment can be get gallium for diagnosis and lutetium for therapy. That is the combination of the diagnostics. So we developed the new types of NOTA and DOTA compound with for gallium and lutetium labeling, and then we tested affinity that KD value for PSMA protein is around 2.5 nanomolar concentration. And also we show the 22RV1 as a PSMA positive tumor cell. And then we also did the blocking study for evaluation of the specificity of this compound. And gallium MSA is good for central lymph node detection and imaging. And also we confirmed that the high liver uptake after the injection of the gallium MSA, and also that the foot pad injection of the gallium compound can clearly show the lymph node in the legs. So especially for a gallium-68 generator, germanium gallium generator is not approved by the FDA, any government body in the world. So they recommend that the DMF2, drug master file 2, type 2 is enough for a gallium generator. So if any other country, especially in Korea, also some company wants to get the FDA approval of that gallium generator, but our Korean FDA clearly show that the generator, especially gallium generator, don't have to get the FDA approval. Just the DMF type 2 is enough for clinical trials. So this is the end of my presentation. So you can get the information of the technetium and also C11 or F18, and also recently developed the gallium-68 compound here. So is there any question or comment for this presentation? Today we have 43 participants in the world. So, 김진실. Do you have any?
네, 네, 교수님.
Do you have any question?
없습니다.
She said no. So this is the last class, especially for the radiopharmaceutical in this semester of the TEIN conference, and also our moderator can share that the information of the chatting box. You should do the fill up the questionnaire in the link, Google Forms. I think you have to go for a lunch. Am I right? So I think we can close this class. Thank you very much, everyone. Thank you.
감사합니다.